Okay, so I promised you that we would talk about the variety of sexual life cycles today. So here we go. I did. Remember, we said there are three main types of sexual life cycles that differ in the timing of meiosis and fertilization from humans. So you have to remember a couple of things before we start talking about these. First, gametes are the only haploid cells in animals, and they're produced by meiosis and they undergo no further cell division before fertilization. So they kind of stop at a point in their development. Gametes fuse to form a diploid zygote that will divide by mitosis into a multicellular organism. But plants and there are some forms of algae exhibit what is called alternation of generations, and this includes stages in their lives where they are diploid and stages where they are haploid, where all of the cells are diploid or all of the cells in the organism are haploid. So a diploid organism is called the sporophyte, and the sporophyte is going to make haploid spores by meiosis. And then each spore will grow by mitosis into a haploid organism, which is called a gametophyte. And a gametophyte makes haploid gametes by mitosis because it's making genetic copies of itself that are identical, that are all haploid. So the fertilization of gametes is going to result in the diploid sporophyte. So you can see here, if you're looking at animals right here, animals have the adult, which is a multicellular organism, which is diploid. Then these cells will divide by meiosis, giving us gametes. The gamete requires the fertilization of another gamete before it will divide. And so it will only start dividing again by mitosis once fertilization has occurred, and you have two sets of chromosomes again. In plants and some types of algae, you'll have a diploid multicellular organism called a sporophyte. Then these cells will divide by meiosis, creating a haploid cell. And then the haploid cell will continue to divide by mitosis, which is kind of a different part of its life cycle. And it will continue to form haploid cells until at some point fertilization occurs and two of the two cells fuse and unite the gametes, so there are two sets of chromosomes again, and that's a zygote, and then the zygote will divide by mitosis, again forming the diploid sporophyte, and then this occurs over and over and over again. So essentially, it looks like there's a diploid generation and then a haploid generation. That's why we call it alternation of generations. And in some fungi, most fungi and some protists, they have a very similar type of thing, the alternation of generation to the plants here. So they will have a diploid zygote, which will divide by meiosis to give you haploid cells, and then these haploid cells will grow into an organism until they are again fertilized. So you can see how these two are very similar. Maybe I just misspoke because I'm half asleep. Okay. Sorry. Alright, so now talking about fungi and protists, the only diploid stage is the zygote. So there really is no multicellular diploid stage. There's just that one cell that is diploid. And the zygote is going to produce haploid cells by meiosis, and each haploid cell will grow by mitosis into a haploid multicellular organism. And then the haploid adult will produce gametes through mitosis. So depending on the type of life cycle, either haploid or diploid cells can divide by mitosis, but only diploid cells can undergo meiosis. So you can't have half a set of chromosomes. That makes sense. So in all three life cycles, the having and the doubling of chromosomes is going to contribute to genetic variation in the offspring. Whoa. Alright, so do you have any questions about alternation of generations? Not right now? Okie dokie. Let's talk about meiosis then, or M-E sperm egg cis. It can also be referred to as reduction division because you are reducing the number of chromosomes through the division of the cell. So it always starts with replication of chromosomes, and then there are two divisions. We have meiosis 1 and meiosis 2. And the 2 cell divisions are going to result in 4 daughter cells, and each cell is going to have half as many chromosomes as the parent cell. So after the chromosomes duplicate, we have meiosis 1, which is called reductional division, and during meiosis 1, the homologs pair up and separate, which results in 2 haploid daughter cells that have replicated chromosomes. And then in the equational division, the sister chromatids separate. So you can think about this like equational as in the equator. So the chromosomes line up on the equator and then separate. The chromatids separate. But the result is going to be 4 haploid daughter cells that have unreplicated chromosomes like so. So meiosis should not be anything new to you. So I'm going to kind of speed through here. Okay, again, I will speed through here. You can follow the color-coded chromosomes. Note over here in prophase 1 that the homologs are crossing over, which also results in genetic variation. So in crossing over, we have non-sister chromatids that exchange DNA segments. So those non-sister chromatids are homologous. Each pair of chromosomes is going to form a tetrad, which is a group of 4 chromatids. And each tetrad usually has 1 or more what are called chiasmata, and they're the X-shaped regions where crossing over has occurred. So if you look back here, see where the crossing over is actually occurring here? That's called the chiasmata. Alright. Cytokinesis is going to separate the cytoplasm, so the result will be our 4 daughter cells with a haploid set of unreplicated chromosomes. And each daughter cell is genetically distinct from all of the others and also from the parent cell. So mitosis conserves the number of chromosome sets, produce cells that are genetically identical to the parent, whereas meiosis is going to reduce the number of chromosome sets from 2 to 1, and it's going to produce cells that differ genetically from each other and from the parent cell. Here you go. See the result? You have 2 diploid cells here, and here you have 4 haploid cells. And here's a summary. There are 3 events that are unique to meiosis 1, and they all occur during the first division, or that are unique to meiosis, and they all occur during meiosis 1, which is the first division. So the first event would be the synapsis and crossing over in prophase 1, and that is when the homologous chromosomes physically connect and exchange their genetic information. So synapsis is when they they physically touch each other, if that makes sense. Then at the metaphase plate in meiosis 1, the paired homologs pair up and divide, which are the tetrads rather than individual chromosomes. And in anaphase 1, the homologous chromosomes separate rather than having the sister chromatids separate. Organisms like genetic variation because it helps the species stay alive, I suppose long enough to evolve. And any type of mutation that occurs in the organism's DNA contributed to the original source of genetic diversity. So if you recall that our theory of evolution tells us that all life originated from a single cell or single-celled organisms, the first genetic diversity occurred simply when there was a mutation in that DNA. So our mutations are going to create different versions of genes, and we call the versions of genes alleles, and the reshuffling of alleles during sexual reproduction is what produces so much genetic variation. Okay. It's the behavior of the chromosomes while the cell is separating during meiosis, and then which cells recombine during fertilization that's responsible for the majority of the genetic variation that we see in each generation. So there are 3 main mechanisms that are going to contribute to the genetic variation that we see in organisms, and those are the independent assortment of chromosomes, crossing over, and then random fertilization. In the independent assortment of chromosome tells us that when the chromosomes line up during metaphase 1, when the homologs are lining up, it's it's completely random how they orient themselves. So it's not necessarily true that all of the maternal chromosomes end up in one side and all of the paternal chromosomes end up in the other side when the cell divides. They can line up with, some maternal chromosomes on one side of the plate and some paternal chromosomes on one side of the plate. So depending on how those sort, each pair is going to sort independently of the others. So depending on which way they line up, that determines the variety of the genetics in the daughter cells. So this is an important number for you to think about and to memorize, this 2 to the n. The number of combinations that are possible when chromosomes assort independently into gametes is 2 to the n. And n is always going to be the haploid number. So in humans, our haploid number is 23 because one set of chromosomes contains 23 chromosomes. So there are more than 8 million possible combinations of chromosomes, because 2 to the 23rd would give you the number of possible combinations. So this is talking about that independent assortment. So you can see here that in this case, all of the paternal organisms, all the paternal chromosomes line up on one side, and all of the maternal organisms, I did it again, chromosomes line up on the other side, resulting in all of these cells having paternal chromosomes and all of these cells having maternal chromosomes. But over here you can see that they line up with one paternal and one maternal chromosome on each side of the metaphase, the equator, the plane where they line up. And as a result, you have maternal and paternal chromosomes in all four of these possible daughter cells here. So crossing over is going to produce what we call recombinant chromosomes. So just recombinant sounds like it's crazy, but the root is just to recombine. So we've recombined chromosomes, and that combines DNA that's inherited from both parents. So crossing over will always begin in early prophase 1 when the homologs pair up. And in crossing over, portions of two non-sister chromatids are going to trade places, which of course contributes to genetic variation because it takes DNA from two individuals and it puts it into one single chromosome. So here's the chiasma where they cross over. This is the centromere of each chromosome. Okay, so the centromere is where the X in the chromosome would be, and the chiasmata is where the crossing over occurs. So as you can see, these chromosomes exchange DNA in two places, which further results in genetic variation in the daughter cells. And then fertilization is random, and that's going to, again, contribute to genetic variation because it's relatively random which sperm can fuse with which ovum. The fusion of two gametes, each with 8.4 million possible chromosome combinations from independent assortment, produces a zygote with any of about 70 trillion diploid combinations. So that, when you do the math, really helps explain why humans look so different from one another. So again, crossing over adds even more variation, and then each zygote will have a unique genetic identity. Of course, genetic variation is going to have evolutionary significance within a population because natural selection is going to act upon the genetic variations, and it's going to favor variations that that persist best in a given environment. So sexual reproduction is going to contribute to genetic variation, which originally came from mutations, and then when the cells divide, they undergo further genetic manipulation, so in crossing over, and then during meiosis, they're going to divide even further. So the point is, all of these processes lead to genetic variation, which influences how an organism will, or how a species will evolve over time.